Hello, 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 everybody. Welcome to the stream. Hello, one, and hello, all. Today we have a very special entry in the Molecular Bio 101 lecture series. But before we do that, let's plug a little something that a lot of you may know that I have been working on for the last a zillion years, this freaking Prion video. So the Prion video, the one that I have been asked to do for many weeks and months, that's finally coming out tomorrow at 10 a.m. So just want to plug that right up front at the start before we get into our lecture today. Today we have a special guest. Some of you may know him in the comment section right now. It is Clockwork. Clockwork, please say hi.
Hey, Vi, thank you so much for having me here. Audience, thank you so much for letting me be a part of this. I'm super excited to talk to y'all about proteins and protein structure. I'm just super jazzed to get back into a lecture capacity whenever I can. I started out my career as a science teacher, but quickly discovered my aptitude was more getting people excited about science rather than instilling real scientific discipline. So I'm always eager to just kind of hang out and give lectures in sort of a space where I'm in a structure like with you, Fi. So I really appreciate you giving me this opportunity here. An audience, after a couple of years, I've reworked my entire channel and we're going to be bringing it back next week. So we're both plugging videos right now. I'm excited for your Prion video, Fi. I'm also excited for my first video back where I do something just obnoxiously ambitious, talking about the eukaryotic replisome. So just excited to kind of take people through that too. But more importantly, excited to talk just like protein basics here and now today. Couldn't be more jazzed for this, dude.
Yeah. Thank you so much for coming on. Yeah, if you don't know, Clockwork, who I want to show off the little avatar that you drew today, which is so cute. Let me go ahead and move that. Clockwork drew his own little Phi character, a little CD4 cell, which is really fun. And if you don't know, Clockwork does some amazing animations for biology on YouTube. He's a fellow biology content creator, and we've actually chatted quite a bit before stream, and we're more alike than I kind of expected, having followed into this I don't know if you call it a hobby or a passion, but this side gig at the very least of proselytizing biology, I guess you could say. So yeah, I'm also very excited to talk basics about proteins. So just a little note in terms of conduct. I've gotten the thumbs up from Clockwork to cut him off if I need to, just because of keeping pace. So if I seem rude, don't worry, we kind of talked about that boundary beforehand. Am I streaming on multiple platforms? Nope, just streaming on on YouTube, sorry. Okay, so today we are talking about proteins, and also let me know if any of our audio levels need to be adjusted. We already talked about DNA on a previous stream, and we talked about RNA, and now we are in the third leg of the central dogma of biology. We are talking about proteins, the thing-doers of biology, everybody's favorite thing to study. Don't at me, geneticists. So let's get started. The Molecules of Life 101. This right here is a special protein that I pulled from PDB, a special bacterial protein. And to begin, I want to kind of reintroduce Clockwork as someone who is passionate about structural biology, because when I look at proteins, I feel like Clockwork has a much higher level of appreciation than I personally do. Even though I find a lot of these protein structures to be beautiful, incredible, and it's really interesting thinking about how they had evolved, I think Clockwork is going to help me sort of bring out the notes and flavors in talking about some of these interesting proteins. But before we get into some of the specifics, let's talk a little bit about protein science as a whole. Right? So here's a little bit about how this stream will go down, this sort of structure. So we're going to start by talking about an overview of protein synthesis. So before we can talk about what proteins do, let's talk a little bit about how we get proteins. Then we'll talk about the basics of protein structures and molecular forces so that we understand how proteins do the things they do. And then we're each going to get a little bit of time in this presentation to kind of nerd out about some of our favorite proteins, to sort of give you an idea of what makes us as biologists excited. And I'm quite looking forward to hearing what Clockwork has to say. This is actually outdated because I'm no longer going to be talking about major histocompatibility complex, and I'll talk a little bit about why later on. And then this is the part where a lot of my concurrent audience really gets to practice their thinking skills. We're going to be talking about how do scientists study proteins of unknown function, sort of the method behind what we do. And then we'll get into looking at actual real data at the very end. So let's just get this show on the road. So I pulled this, I believe Clockwork, you gave me this GIF. Do you want to explain a little bit about what this is?
Definitely, and just annoyed at PowerPoint for not having looping GIFs. Oh, it is looping. Awesome. So this is one of those fundamental structures for all life on Earth. I would love to also talk about the structure you picked for the cover slide too, Phi, because it has an actual really cool part of history. That's the injectosome from a certain bacteria. And this is actually similar to a structure that was kind of used in a legal argument to sort of prove that evolution like is legally real and to sort of like quash intelligent design being taught in schools back in the aughts. But to get back to the actual protein on screen, this is ATP synthase. Protein structures are amazing because of like their complexity, but the ultimate, like when you get down to it, if you kind of break down what they actually do, it becomes things that are like super similar to what we see in everyday life. Like ATP synthase is what takes protons from within your mitochondrial inner membrane and uses them as a literal like pressure gradient to put ATP, the cellular energy currency, together. And so like we, a species that eventually went on to build the water wheel and like electromagnets that can be used as motors, are powered by a spinning turbine, basically. And I think that's one of those things that like really kind of like blew my mind in terms of I have to just study and talk about structural biology. So this is one of those things that might be a little bit too complex to explain all the ins and outs of how ATP synthase works on this stream. But what I'm excited about how you've structured this lecture, Phi, is how we can kind of level up into understanding how genes, DNA can turn into something this complex and something that works as well as it has for billions of years at this point. And
Yeah.
just sort of, like, examine the pressures along the way too.
And if you want to learn more about ATP synthase, it's, I think, one of the best videos on ATP synthase on here on YouTube is on Clockwork's channel, where he goes real hard on making sure you actually understand, even if you've never taken a biology class before, actually understand how proteins work, which is just insane. Okay, so let's talk a little bit about the central dogma of biology. So if you are a biology student, then this is something that should be essentially written into your brain at this point. But if you aren't, this is the one thing that you should take away from your Bio 101 class in high school. It's that DNA is the blueprint of your life, contains the data that codes for the stuff that make you you. And then that is read to make this intermediate called RNA, and then that RNA is read to make protein. And so this is the central dogma. It's the starting place of molecular and cell biology. And the process by which we go from DNA to protein is called translation. So for DNA, you undergo transcription to form RNA, and then RNA gets translated into protein. So proteins are the thing doers of life, and we're going to ignore RNA because I already talked about RNA. RNA can do things too, but proteins are the main thing doers of life, which is why they are so interesting to talk about. Certainly, if you work anywhere in cell and molecular biology, you will have to think and talk about proteins. That's how essential they are in our discipline. So just to quickly go over how proteins are made, they are made block by block. So they are made of strings of these things called amino acids. And so these amino acids are stitched together within a structure called the ribosome. The amino acid building blocks are brought to the ribosome by these hybrid RNAs called tRNAs that are attached to these amino acid building blocks. And so these get stitched one by one into a growing peptide chain. And so that's just what you need to know on how proteins are built, but that's going to allow us to understand, knowing what proteins are made of, a lot of interesting chemistry and physics that come out of understanding these basics. So proteins are built from about 20-ish amino acid Lego blocks with differing chemical structural properties. So there are the nonpolar uncharged. So these are amino acids that don't contain that big of a difference in charge polarity. You have the aromatics, which are characterized by these ring structures. You have the polar uncharged amino acids that have a slight difference in charge at either end. And then you have the amino acids that sometimes carry actual real charges, positive and negative. And so each one of these amino acids, if you follow me on Twitter, I'm at neutralphil. I made these memes where I gave each of these amino acids like personality and asked you to tag yourself. Because I think each one of these does have their own sort of contribution to structure. Clockwork, do you have a favorite amino acid out of curiosity?
I don't know why, but like I always saw these warnings about phenylalanine, like in various like food products, and so I just kind of like it was like the first amino acid I recognized when I was studying biology. So I always just have like had a soft spot for phenylalanine. But in terms of like studying structural biology and being like air quotes excited when I see a residue, it's tyrosine because of just all the different ways that tyrosine be tyrosining. But I don't think we're gonna get into that in this stream. How about you, Phi? Like, based on like a much richer background, has a sort of favorite emerged on your end, dude?
I don't think I have a richer background than you do. You're much more of an actual structural biologist than I am. But I have to say that I'm partial to glycine as being a weirdo. And as Surprise Pants has noted in chat, we will talk about these weirdos later in the stream because they have some very interesting structural properties. Yeah, so I'm partial glycine because I like. Maybe I'll talk a bit about it when we talk about glycine as a whole. So, understanding that these proteins are built out of these 20 unique personalities is going to allow us to understand how proteins are formed, why they take the shape that they take, and why they do the things that they do. So, keep all of that in mind. Yeah, and we're going to talk about the weirdos. That's why I have this box highlighted. So before we talk about the weirdos, it's important to understand that there are a couple of different levels of protein organization, from primary to quaternary, and I'm going to have Clockwork kind of join me in discussing quaternary structure, but I'll go through the first three just because they're pretty straightforward. So primary protein structure refers to just the identities of those amino acid blocks in the order that they are stitched onto the protein, right? So this is a very simple peptide. It's got sidedness, so N termini and C termini. And so you can go down the line and see which amino acids are stitched in what order. So methionine, phenylalanine, I think this one is aspartame, aspartic acid, maybe. I used to know all these, and if you were ever studied biology, you had to memorize all these, but those charged ones, I always get confused. So you can describe protein structure at that initial ordered level as well. Where it gets kind of more interesting is this secondary structure. So secondary structure can be broken down into two basic motifs that this primary string of amino acids folds into. So you have structures like the alpha helix, which kind of reminds me of the structure of DNA, but they're not quite the same. They're both helical, but the alpha helix is slightly different. And you also have these beta sheet structures. And if you are looking forward to my prion video, understanding that proteins are made of these motifs, alpha helix and beta sheet, is actually going to be really, really important for when you watch my prion video, which, again, plugging myself, goes up tomorrow at 10 a.m. EST. It wouldn't be very interesting if proteins were just at this level of organization, so they also fold into these tertiary structures, of which I am covering right now. So I'm going to go ahead and kneel for a hot second. And so these strings of beta sheet and alpha helices are all strung together by flexible chains of amino acids, and they fold into these interesting patterns that give proteins their characteristic shape. And I'm going to go ahead and let Clockwork describe quaternary structure, because this is going to be important for our discussion of hemoglobin. So yeah, Clockwork, why don't you take it away?
So I'm right here, and what gets really interesting is tertiary structure is, we're going to get into the things that cause tertiary structure, how proteins kind of like coalesce into these bigger three-dimensional shapes based off of beta sheets and alpha helices. But the cool thing about quaternary structure is a lot of proteins are actually made up of individual subunits of different tertiary structures. One protein we're going to talk about, sort of like leveling up through here, is hemoglobin, which is actually 4 different subunits. If you take each of hemoglobin's 4 subunits and just kind of look at them individually, they're basically just a myoglobin particle, which we'll get into how that sort of changes what the protein do. But you can kind of think of, if I did a great job organizing this too, because you can kind of see that like each individual monomer forms a dimer or a trimer, right? And so you see these interesting ways in which the geometry can form. Like sometimes you have radial symmetry. Sometimes if you go back to ATP synthase, you have things like an actual sort of ring of columns. One thing you see a lot too with a lot of quaternary structures is transmembrane proteins with 6, 7, or 8 how alpha helices hanging out in a very specific orientation, and it's important how those kind of interact with each other to sort of form signal transduction. So
Yeah.
tertiary
So
structure is how two secondary structures interact, and quaternary is how tertiary structures interact to form sort of like a full protein unit, if that.
yeah, I like that organization. So primary builds into secondary much the same way as tertiary builds into quaternary. So quaternary, in terms of symmetry, we have a sample protein, and you can kind of see here that each one of these units is called a monomer, and then when you have 3 of them together, it's called a trimer. So as we have in the comments slipped remarking that quaternary is quite beautiful. Yeah, a lot of proteins are really interesting to look at, and it's part of what makes understanding structural biology so amazing, and we'll get into a little more of that in the future. Okay, so that's a pretty good introduction to protein organization. So now we have the vocabulary to discuss what it means to study proteins. So yeah, we have the vocabulary. So let's talk about protein folding. And again, this is such a good teaser for the prion video because prions are all about folding, and when prions misfold, a lot of terrible things happen. And so it's important for us to understand the forces that govern how a protein actually folds. So as we saw in the previous slide, proteins are just strings of these amino acid building blocks, and they fold in very specific ways. But it's important for us to understand why they fold in the shapes that they fold. And so what governs this ability to form these interesting structures? So secondary structures form what are known as intramolecular bonds. So these are bonds in between these specific molecules, right? So if we zoom into a beta sheet, this is of course just a cartoon. These proteins, these amino acids are not actually arrow shaped, right? These are cartoons to give us an idea of what they look like at the molecular level. But if we zoom in and sort of remove this illusion of the cartoon, we can kind of see that there are specific bonds, these dotted lines, in between the hydrogens and oxygens that make up the amino acids. So if you're a biology student, then of course you're going to go into much more detail when you learn about this for real. But if you're not a biology student, then what you need to know is that R's are basically the parts of the amino acids that make them unique. And then this part here are just elements of the amino acids that stay the same. So this oxygen binds this oxygen binds this hydrogen. And it's these electrical forces that holds the protein together at the secondary structure level. So that's how you get these patterns, right? So there's this arrow that goes to the left and the arrow that goes to the right, and we simplify that with a cartoon like we do here. I think what's more interesting though is talking about tertiary structures, so how these beta sheets and alpha helices fold. And I want to ask chat right now, and if you have a education in biology, then hold your horses, don't spoil this for the non-biologist in chat, but I want to interrogate chat or to put chat on the spot and really get the mind thinking about how proteins fold. And proteins fold in their tertiary structure somewhat dependent on the environment they're in. So can I get an answer from the non-biologists in chat or the non-chemists in chat about the environment that proteins form in? What's a major component of the environment that proteins form in? You see electromagnetism, that does have I guess it's somewhat correct. Give us choices. I think this one is more interesting if I don't give you choices. So we have Mr. Fox saying water. We have Slipta saying temperature. A saying heat, hydrogen bonds. Certainly all good answers. And so, and Kevin says acidity. These are all really good answers. However, Mr. Fox is the one that has the answer that I was thinking of. It's water, so our friendly H2O. Now, the rest of you are correct. Acidity, or in other words, the concentration of hydrogen ions in solution, that does affect how proteins fold. Temperature definitely affects how proteins fold, especially if we're talking about prions. But the thing that I want to talk about right now is water, and why water is so important. And often, if you take a college level biology class, you'll often spend a whole day just talking about water. So water has this property where one side of the water molecule is partially negative, and the other side of the water molecule is partially positive. And it's this molecule that has this inherent polarity, meaning having poles like North Pole, South Pole, that allows us, that forces proteins to form in a certain way. Water is hella weird. Yeah, it really is, from a chemical perspective. So a major molecular force that causes proteins to fold in the characteristic shapes that they fold in is what's called the hydrophobic effect. So as you've seen, I'll just tab back to the amino acid chart. There are proteins with various charge properties. You have the nonpolar uncharged, so again, amino acids that don't have a positive and negative side, but then you also have amino acids that do have a positive and negative side. And so the hydrophobic effect states that amino acids that like water, that have this charge, will be on the outside, whereas amino acids that don't have that charge or charged polarity like to hide on the inside. And that actually allows you to get a lot of the way through to how proteins actually fold. So in black, in this cartoon polypeptide chain, we have amino acid residues. Another word for amino acid is just a residue. We have in black the hydrophobic ones, so the ones that don't like water, the ones that don't have charge. When you make that protein, it will fold in a way where those amino acids that don't like water will hide in the center of the protein, form that core, whereas the amino acids that like water will be on the outside. Okay. So, just as an intuition check, chat, does this rule hold for every amino acid in a protein? Could you ever find a hydrophobic amino acid on the surface of a protein? There's no way that, unless you had a biology education, that you would definitely get this right. But just as like a way to engage in the kind of idea of protein folding. Does every amino acid on the outside have to be hydrophilic, loving water? Give you a second to think about that. And I'm kind of curious, Clockwork, as to why you think I put this question in.
I'm trying to like game it out and figure out what concept you're aiming at, and I'm drawing a blank. So I'm gonna just keep a even keel and see where you're going with this. Very excited to see, but drawing a blank right now specifically.
Yeah. So what I want to get across is that at a basic level, a lot of biology and chemistry does not have hard and fast rules, right? Certainly there are mathematical rules that things abide to, but something like this, proteins are such long chains of amino acids that there's almost always going to be exceptions, as Slipta had said. So these are trends. They don't hold strictly true in every single case because of things that are local to that structure. So just expanding your mind to think of these not being hard and fast rules, but rather tools that we can use to explain natural phenomenon. Will the stream be safe for later rewatching? Everything is archived on YouTube, so no need to worry about that. So let's talk about weirdo amino acids. It's about time. We know a little bit about how proteins fold, so each amino acid has a particular characteristic by which it contributes to how a protein is structured. And I call them the big three, the big three weirdo amino acids. And so glycine, and here's where I'll also call upon Clockwork's expertise in case you have anything to add after I give my spiel, because these set of three, these are my favorite amino acids just as a whole because of how weird they are and how they contribute to protein structure. So glycine's R group, which is the part of the amino acid that is unique to that amino acid, is just a simple hydrogen, making it a floppy link on the chain. So other amino acids that we're going to see have a lot of structure, and that structure constrains the flexibility of that protein. So if we look at this figure, we can see that there are these molecules. This one's a nitrogen in blue, and in red, these are oxygens, and in gray here, we have carbons. These links here are actually somewhat flexible. They can bend and rotate to a certain extent. Glycine's R group makes it really floppy, so it has more of that flexibility and is less constrained. Actually, I think it has less steric. It's less bulky, so it doesn't get in the way of other amino acids. So you'll often find a lot of glycines in parts of the parts of the protein structure that are very floppy and have a lot of flexibility. Clockwork, do you have anything to add about glycine?
And that floppiness is absolutely essential because we talk a lot about why proteins fold, but a thing for me, as somebody who looks at all this stuff, is also why do proteins change shape and what causes that stability? So while talking about glycine's cool, I am chomping at the bit for you to get to the next weirdo amino acid, which is one of the real good ones in terms of conformational changes.
Yeah, so let's just get right into that. So glycine is the first weirdo one, and then we got cysteine. So cysteines R group can form sulfur bridges. So this yellow ball here is a sulfur. And so this sulfur molecule can form what is called a disulfide bond. So if this cysteine finds another sulfur, it can actually form an actual chemical bond that's more than just a hydrogen bond. So not intramolecular, but intermolecular. So these amino acids can sort of staple proteins in a fixed way, which is a really drastic way to force a protein to act in a certain manner or have a certain structure. So I'm excited to hear, Clockwork, what you have to say about cysteine.
It's just one of those things where a lot of the chemistry you see in amino acids is very malleable in sort of the water environment. We talked about how proteins exist primarily in water, and so things like polarity, like hydrophobic, hydrophilic, and charges. Since water is a charged interface, right? Those can get messed up pretty readily by just the ambient environment. So one thing that's cool about cysteine is that disulfide bridges, they're not stronger, so to speak, but they're harder to mess with based on what's going on in the ambient environment. And so that's what makes proteins so powerful, is that there's so many different kinds of interactions that if you sequence them in the right way, you can create just stunning levels of folding and therefore stunning levels of functionality. Like watching how this all interacts with something as complicated as how your DNA is dividing right now, trillions of times a second, is totally bonkers to me. Just all the different intermolecular interactions that just to prime your DNA on the lagging strand. I don't want to get too deep into esoteric stuff, but that's the thing I want to really kind of hammer home here, is that all these different interactions are amazing individually, but it's that whole they add up to that creates the incredible complexity that is biochemistry itself. So cysteine rocks because it's one of these things that you don't really see, air quotes, that much in organic chemistry or Chem 101, which a lot of you might be going through. And therefore, in sort of the aqueous environment that is like the cytoplasm, they provide an interesting place where conformational changes can both happen and then be stabilized. But again, I don't want to get too deep in it, just glossing over.
The other thing about these bonds is that, I mean, is it actually true that these are not stronger than other bonds, Clockwork? Because don't they survive boiling?
In some cases, they do. I didn't want to introduce a ranking into them immediately, right? Because it's not necessarily oh, how strong the bond is. What does the interaction contribute to the overall shape of the final dimer, trimer, or whatever? You know what I'm saying?
That's fair. We might not want to get dragged down in that in a ranking system. That's fair. Okay, let's talk about the last of the weirdo amino acids, proline. So proline is a straight up freak. So if we look at the structures of glycine, So this is just the hydrogen that pops off. Very simple, very elegant. Right here, we just have this additional sulfur and carbon group. The proline's R group actually connects back onto the backbone as a loop. So if you can imagine each one of these links being somewhat flexible, well, if you loop it back onto itself, you've created a high level of inflexibility. So you actually don't maintain that rotational conformational flexibility. You actually form a rigid kink in the chain, which is why I have in that Twitter post proline being the kinky amino acid, because it is a little bit of a freak. But we love it all the same. Okay, so that's basically me. Did you want to say anything about proline or?
No, that's basically it too.
Yeah. It's cool. Proline's cool. So let's get right into the next segment. So 100 proteins do stuff. Too long didn't read its chemistry. Biochemistry will actually be a different 101 lecture. Otherwise, it would take far too long. But just to give you a little bit of a taste of how these proteins do the thing that they do, all of these amino acids, some of them have interesting electrical properties. And those electrical properties in those R groups can catalyze chemical reactions. So here we have a classic catalytic triad consisting of a acidic amino acid, a basic amino acid, and a what's known as a nucleophile. I don't want to get too deep into it, but these catalytic triads can help electrons move in different ways, and that it will allow chemical reactions to be catalyzed. So these specialized proteins that catalyze chemical reactions are called enzymes. So if you weren't aware, enzymes are proteins, but not all proteins are enzymes. So we'll get into that in a future lecture, but just wanted to talk about proteins for today. So we're going to get into, we're going to get into a protein story, and I'm actually going to let Clockwork take the wheel on this, as I asked Clockwork to provide a protein that really solidified his passion for biology, to illustrate and kind of vamp about what makes certain proteins so cool. So Clockwork, the stage is yours. What do you have to say about hemoglobin? And let me know when you want me to progress the slide.
I will. I'll let you know. I'll just do the classic next slide thing. So very excited to sort of get from the not theoretical, but from like the basics into how this stuff actually works in the wild. We are still going to only kind of cover like the very introduction to what makes hemoglobin so cool, because hemoglobin is actually one of the most studied proteins in history. If you look at this right here, this is two hemoglobins kind of like rotated. You're looking at a 4 subunit protein. So that means that its quaternary structure is just stapling together 2 alpha globin proteins to 2 beta globin proteins. So they're a little bit similar, a little bit different. And the main sort of feature of hemoglobin is that each of these subunits holds on to something called the heme group, which is basically a big old ring that's surrounding an iron atom. And so these guys hold on to iron basically. And what's that iron for? We're gonna find out. But the thing for me that's cool to kind of level up through this is kind of talking about how do we even know anything about hemoglobin in the first place? If we can just get to the next slide, we'll kind of roll through these very quickly, right? The main thing about hemoglobin is it's actually very easy to study, even if you don't have like cryo-EM and all like the fancy stuff that we built in the 20th century, because we've been able to study like individual components of blood for a long time. To kind of go way back to it, hemoglobin is in your red blood cells. Red blood cells are, the thing that makes your blood red, not to get too sort of like with it. And if you have a centrifuge or anything you can spin really fast, you can actually separate all of the red blood cells out of your blood plasma and then just kind of do whatever you want with them. People in blood banks separate them out to get basically more oxygen carrying units from anybody who's donating either just plasma or lots of other things. But back in the day, in literally like the 1850s, people were spinning down blood and just seeing, what it was made of. If we go to the next slide, we can talk about what red blood cells are. They're kind of one of the weirdest cell types we have in our body, because unlike most other cells, they don't have a nucleus. It's kind of a head empty situation. They're just big bags of hemoglobin, right? So what you can do is you can kind of break that cell and then study what's inside. And so as people began studying and understanding what makes up red blood cells, what we have is just something that is these like little crystals, right? So people were isolating hemoglobin, finding the iron in it, and discovering its oxygen carrying capacity as early as 1875, right? But until we had like cryo-EM and the ability to sort of see things at a molecular level, we couldn't really understand how hemoglobin carries oxygen and why it do that. What's even going on there? One really cool caveat that I want to point out real quick is that hemoglobin, as we were studying biochemistry, as we were studying sort of like the organic makeup of human anatomy and physiology, it's not the only compound we found that carries oxygen and carries oxygen well. There's also in your muscles a molecule called myoglobin, which is going to form an interesting way to compare how proteins do their jobs by comparing exactly what hemoglobin and myoglobin do and why they do it. So now that we're getting into the actual workings of what hemoglobin is, that's the main thing. Hemoglobin carries oxygen, but what does it actually mean to carry oxygen? And just real quick, Phi, can you come in and can we talk? Can I have you sort of like check me with talking about binding chemistry? Because I don't want to go too deep into like the biophysics here.
Yeah.
How do you want to like cover this real fast?
So I have this slide here, and I'll actually maybe do this because I put this slide in at the last moment, because I realized that the way that a biologist or a chemist thinks about binding is it's not exactly the same way that you might think about binding traditionally, right? So this is my binding chemistry speedrun, any percent. I mean, I said we wouldn't talk about biochemistry, but we kind of have to, just a little bit, right? So
just a taste, just a taste.
Just a taste, right? Biology is just molecules bumping into one another. That's all biology is, simplified at a very basic level. And unlike macro level materials, molecules bind and unbind at some ratio, right? So you have rates that describe how fast two molecules like to bind, and then you have rates describing how molecules like to unbind. And so there is this ratio of binding and unbinding that gives us an idea of how strongly something binds. So chat, as a quick check for understanding, I'll give this a poll here. So if a protein, how do I want to do this? It's kind of a last minute addition. Maybe I won't do a poll. But in any case, macro level structures, right? Say you are gluing two pieces of paper to one another. You apply the glue to one piece of paper, and then you stick those two pieces together. They're going to be binding forever, more or less, right? But on a molecular level, these interactions are not permanent, right? There are going to be molecules that are binding to one another and then molecules that are falling off of one another. And so the important thing is, when it comes to understanding binding, is that the tighter something binds, the larger the ratio between these two states actually is. And I'm trying to, maybe that's not the best explanation, but I think that gives you an idea. Things unbind and things bind. They don't just stick together permanently, no matter how strong they are, I guess you could say.
Can I take a shot downfield here, Phi, and you can just check me? We really want to be in a situation where things are binding and unbinding in equal ratios, because if anything comes in and binds too strongly to our biochemical molecules, they're not going to be able to go back to their original form, which means you're going to have all sorts of problems. Hemoglobin, for instance, super cool how it can bind and unbind with oxygen, but if it comes in contact with, say, carbon monoxide, that has a much higher, tighter binding affinity, and that's going to bind to your hemoglobin and not let go, basically, and that's bad for you staying alive. Is that a good way of thinking about it? Can I use that metaphor?
Let's actually look at a hemoglobin in the next slide so that we can actually see kind of, and so the reason why I wanted to do that as speedrun concept is because we're going to be looking at binding curves. So this is a very exciting feature of my streams where, we not only want to do a 101 lecture that anybody can enjoy, but really challenge people. And so I think looking at data is interesting and worth trying. Even if you don't fully understand what's going on, it's nice to be able to see the math behind all of this science. So with our understanding that things don't just stick together permanently, we can now kind of understand what these curves mean. So Clockwork, why don't you take it away here?
And that's what I want to get into as well, is talking about how we were able to figure out what these molecules do without having sort of like the really powerful microscopes necessary to really fully understand like the mechanics of how these things work. Like scientists looked at these two curves on screen right here and were like, oh wait, I think I understand what hemoglobin does, and I think I understand what myoglobin does. I'm not going to give you a quiz on this or anything, but I want to kind of guide you through this data and see if we can figure out like the difference between hemoglobin and myoglobin and try to figure out what their jobs are. Because while both of these guys interact with oxygen, they interact with oxygen in very different ways. It's very important. So I want you to look at the graph on the left first. This is hemoglobin's binding curve. If you start at the bottom left, the x-axis is the amount of oxygen in any given environment. It's the percent O2 based on pressure, so the oxygen pressure. And then on the y-axis, you have how much of our hemoglobin is saturated, right? Is it 10% or is it 100%? So in an environment like at the bottom left, it's kind of the obvious answer, right? When you don't have a lot of oxygen, hemoglobin's not going to bind to oxygen. But what's very interesting is, as you go through this curve, rather than like it being a straight line, there's two kind of states where in the middle, it starts out pretty slow and then ramps up and then begins to level off, actually. So once hemoglobin hits a state where it's in an environment where it's at 70% oxygen, it's basically going to be 100% bound to oxygen. So at some point during this curve, hemoglobin actually like accelerates its ability to bind to oxygen. So the thing I want you to look at is these different numbers right here. I want you to look at hemoglobin at 40. So at 40% oxygen pressure, it's about 70% bound. At at 60, it's a little bit over 80, and then at about 80%, it kind of tops off at 90%, and that's pretty natural for a binding curve. You're not going to see something hit 100%. Once you see the level off, that's where chemists stop being interested, right? If we move over to myoglobin, though, it's way more pronounced. Like there's no S curve here. It's just up and instantly goes to the right. So something interesting here, myoglobin will start binding to oxygen super fast, as low as like the 30 level. Once you hit 40, it's kind of over for myoglobin, whereas if you go to 40 on hemoglobin, it still has some binding affinity left in the tank, right? And that's where we kind of begin to understand what hemoglobin does and what myoglobin does as well. So I don't think this is enough to directly ask the question, but Fi, if you'll be patient with me, I kind of like, let me just ask you, Fi, you look at these two things right here. I know you know what hemoglobin and myoglobin do, but if you look at these curves, like what would you guess like the actual roles hemoglobin and myoglobin play are? Like how would you think about their sort of jobs if they bind to oxygen in this interesting way?
Yeah, so I'm going to answer this in a very sincere way, just because I do sort of know the answer. But from what I'll say, like from a naive perspective, it looks like myoglobin is really, really good at binding oxygen when it's not very present, and that I think is obvious from looking at the curves without understanding anything too deep about chemistry, right? If we look on the right, as you said, at very low levels, myoglobin is very good at picking up that oxygen, but hemoglobin kind of looks like it sucks picking up oxygen for a while. And so this difference in how they pick up oxygen is, I think, that's kind of what I would note if I didn't know the answer behind the scenes. And so I'm guessing there's some difference in the environment that they're needed in, is what I would say.
Exactly. And audience, one thing I want you to do too is I want you to look at the level of 40 at the x-axis. So on the bottom there's a 40 in the hemoglobin chart, and at the bottom there's a 40 in the myoglobin chart. Look what happens when you move to the left of 40 in hemoglobin, and then look what happens kind of drastically if you move to the left from 40 in myoglobin. And you can start really wrapping your head around this. Again, I'm not expecting you to put it all together based off of this. I'm literally cutting out 3 lectures worth of information to get you here. But let's see if we can't add another dimension to this. If you could skip 2 slides ahead, because the slide in between these is just the notes between these 2 slides.
Before we move on, though, there are commenters asking what DPG is. Do you have like a two-sentence explanation for those with them who are curious?
So what I really want people to focus on, what I did with these charts is I took from the end of like a 4-lecture series, right? So I really want you to focus on just pH instead of thinking about DPG is. Just so we don't get confused here, just know that pH moves the binding curve a little bit.
Gotcha. So basically, don't worry about DPG.
DPG literally stands for "don't worry about it."
Okay, cool. Okay, so I'm skipping 2 slides ahead. All right, I'm
Perfect.
there.
And if we think about sort of the levels at which we see, if we think about areas of the body where we see these oxygen pressures, we can begin figuring out what the actual roles are here. So I want to really point out this is a huge average. It's never directly like this, but you can kind of get a sense of what's going on if you see sort of different oxygen pressures throughout tissues in the body. In your lungs, you take a deep breath in, you're going to hit, a good 80%, like a good over 80 percent oxygen in sort of in terms of pressure. In average tissue, it's hanging out at about over 40. Like on average, your like cellular structures are operating at about half the amount of oxygen that are in your lungs. So you're taking a deep breath. What's really interesting is in exercising muscle that gets basically cut in half again to around the 30s, even down to 20, right? So if we kind of think about going back to that first series of charts and thinking about that 40 moment, we can kind of really see what those actual roles are. What I did here too was I didn't actually have the two axes on the same side because I wanted you to really think abstractly and rotate this in your head. I didn't want to make it too obvious. So we know hemoglobin's in our blood, but if we look at those levels of oxygen and we look at that binding curve where we know the actual role of any one of these things is actually to let go of oxygen, where do you think? And anybody who knows Latin or Greek, don't answer. But if you could guess this, we know hemoglobin's in the blood, but if you look at this binding curve and then look at where we see these different levels of oxygen in the body, where do you think myoglobin like hangs out the most? Where do you think you're going to find the highest percentage of myoglobin in the human body?
Yeah, so that's so take a look at the myoglobin binding curve here, where it's really good at binding at low levels of oxygen. And then we can head. Oh, I mean, chat's already got it. I thought they might have needed to tab through some of the different charts, but yeah. So myoglobin's really good at hanging on to oxygen at low levels of oxygen, which, as
Yeah.
the chat has correctly described, is where myoglobin hangs out. And as you teased, myo meaning muscle.
Yeah,
Very,
I didn't
very
know.
cool.
Oh, as somebody who was raised like in an extremely Catholic environment, I never know how many people are like fellow like Latin/Greek heads. So I say you can't cheat by using knowledge you may already have, and that kind of gets to the core of it too. It turns out that myoglobin is much better at binding to oxygen. Like it's going to hold on to oxygen atoms unless like there's just nothing around. Whereas hemoglobin kind of is terrible at binding to oxygen, but that's what makes it really good at its job. So if we can skip two slides ahead, again, we'll skip past to just like the way we can start putting this all together. We can kind of see exactly what's going on in terms of why this is important. So myoglobin is really good at binding to oxygen, which means that the only thing it's actually good at is storing oxygen. So you dump a bunch of your myoglobin into your muscle cells because it's actually very hard for your body to get oxygen to your muscles during sort of the insane effort it takes just to even stand up and walk around. Like the amount of energy it takes to move a sarcomere, which is an individual chemical unit of a muscle. Don't get me started. The amount of energy there is wild, and therefore you need just a ton of oxygen just to like even walk around, right? So the thing that makes hemoglobin really interesting is it's actually very bad at binding to oxygen outside of certain very specific instances, and that's where quaternary structure comes into play, and that's where the most important part about proteins comes into play as well, and that's conformational changes. Hemoglobin actually changes its shape. If we can go to the next slide, we can begin understanding exactly what's happening here. And this is why hemoglobin for me is such an awesome introduction to what biochem is, because if you are on this slide right now, what you're looking at is on the left, you're seeing sort of a air quotes molecularly accurate representation of hemoglobin, which is honestly a little bit hard to see. And then on the right is sort of a chart of what's happening. So in the lungs, hemoglobin is able to change its shape a little bit to be more able to bind to oxygen. And then as it goes through the body, it's going to encounter different pH levels. And the thing about cellular respiration, the way that you turn food into energy, actually, you take oxygen in and you bring CO2 out, right? You do whatever you're going to do to create energy. I don't want to get. I would love to talk about the Krebs cycle right now, but I know you don't want to. um, but like the equation is food plus oxygen equals water plus CO2 at its most basic point, right? So anywhere your body needs oxygen, CO2 is going to be there. And the cool thing about carbon dioxide is that it dissociates in water, becomes acidic, and so anywhere you're going to have sort of a lower, more acidic pH, you probably are going to need oxygen there. So as soon as hemoglobin that has oxygen on it encounters a low pH situation, its shape is going to change because pH will interact with some of the salt bridges and some of the van der Waals forces within this quaternary structure and allow it to change just a little bit. And that shape change, if you watch at the end of the GIF, you kind of see oxygen when it pops out. You see how it like allows the heme group and the iron to come out a little bit, and that allows oxygen to make its escape and go down its concentration gradient. And for me, that's kind of the thing that like really seals how cool proteins are, right? Because the structure is so fine-tuned by evolutionary pressure that hemoglobin does its job beautifully. It's not there to hold on to oxygen. If it held on to oxygen too tightly, it would be terrible at its job. You would, not be alive, right? But every breath you take is very well distributed throughout your body because as hemoglobin goes down its oxygen binding curve, it releases the exact right amount of oxygen at the exact right time. Now, to kind of take you to like the next level there real fast too, we talked about quaternary structure a little bit. The thing that really seals this is in the lungs, hemoglobin actually binds oxygen sequentially. And what that means is that pH can change the shape of a protein. Heat can change the shape of a protein. Looking at a protein funny while you're doing a cryo-EM micrograph can change the shape of a protein, which is very frustrating if you're in a lab situation. But also, the shape of the protein next to you can also change your conformation. So that's why quaternary structure is so important. Hemoglobin when one hemoglobin binds to oxygen, the odds of every other subunit within that hemoglobin binding go up drastically, which is why you see that curve level off, which means that as hemoglobin is going through the lungs, it's going to bind to oxygen very quickly and then hold on to it. There's another part of this. There's like a level 3 to this that I don't want to like take up 2 hours of your time or anything. That makes it really fascinating why hemoglobin doesn't immediately just rebond to that oxygen. But I don't want to talk about how that shape stabilizes because I feel like that's very much a 201, 301
Yeah.
situation to talk about. But any questions from you, Fi, in terms of like why that's important or anything else I can kind of hammer out in terms of why quaternary structure is important and why these binding curves are important as well?
Yeah, so let me make an attempt to summarize from my perspective why this is cool. Myoglobin's made of one subunit, and so it has this very simple binding curve. It sucks. It's really good at binding when there's little oxygen. What makes hemoglobin different is that it is made of 4 subunits, which influence each other's ability to bind oxygen. So again, I agree with you're talking about cooperative binding, which is definitely a 201 kind of idea. But essentially, because hemoglobin is made of interacting proteins, it's allowed to have this more complex shape, right? This shape where it has a couple of different phases that you can clearly see. And this flexibility, this conformational adaptation, allows it to suck at binding oxygen at certain instances and get really good at binding oxygen in other environments. And I agree. When I learned that in biochem, it was actually mind blowing. It was really, really interesting. Yeah. Is there anything else that you wanted to work out on these slides, Clockwork, or Clockwork, you there?
Oh, I was on that hardware mute. No, that's terrible. Huge. We're talking like a basic mistake. How dare I? Very embarrassing. No, but in terms of covering this, obviously, as somebody who is fascinated by hemoglobin, the answer is yes. But in order to cover sort of like the next levels of what affects hemoglobin's binding curve, we'd need 2 to 3 more hours. So, Chad, if you really want to hear about that, I just need you to bully Fi into letting me come back, and we can like do a biochem like 201 style situation where we can talk about. But if you want to learn it yourself, I want you to type in the phrase 2,3-BPG hemoglobin into Google and see if you can't make what you will from there, but not right now. Stick with the rest of the lecture.
And subscribe to Clockwork, obviously, because he goes to not extreme, I don't want to scare you off, but he goes into quite crunchy, delicious details about how proteins actually work, whereas I care more about the functionality of the story. So it's kind of an interesting difference in what we find interesting, which is very neat.
And to be fair,
Yeah.
I find what you talk about to be way more interesting. It's just that I have a hard time picking things to talk about, and for me, biochem's just a good place to focus, right? Like if I was going to do a prion video, I'd accidentally end up talking about different deer species for 3 hours and then talk about prions.
So shall we move on to the next story, or was there anything left here?
I think that's a really good place to leave it because you
Okay.
begin to see, how these things work. And your protein story, frankly, is way more interesting to me. So I'm just super jazzed to get into it.
Well, interest is relative. I think for a lot of people who study proteins, hemoglobin is like their first crush. Let's talk about C3, okay? So my story is about C3. C3 is a protein of the immune system, and it is part of what is called the complement cascade. So the complement cascade is a weapon that the immune system uses to blow up bacteria and other cell-based pathogens, right? So you don't have to know each of these individual steps unless you are going to medical school. But the idea here is that C3 is one of these proteins that contributes to the formation of what is called the attack complex that we see down here at the bottom of the slide. So the attack complex is this barrel that essentially punches a hole through bacteria, and it's badass, and C3 is one of the proteins that contributes to the formation of this bacteria-busting protein. That's already cool. I think for immunologists, that's one of the first loves in immunology, is understanding the attack complex. We aren't the complement system, we're the insult system. No, chat, please don't. So C3 is a component of the complement cascade, but my taste in biology comes from stories that are unexpected. So this protein that people understand quite well in the immune system, how this actually functions and forms this attack complex, actually has a very, very different function in the nervous system. So C3 plays a completely different role in neurons, completely different role, right? C3, and I want to take a little time here so that we can really appreciate this problem, right? The immune system and the nervous system, for you fellow boomers out there, were thought to be completely separate. And this is a point that I've hammered home in my rabies video plenty, where when we were growing up, we were told that the immune system and the nervous system don't interact because it would be really bad if you had inflammation in the brain, right? If you had inflammation in the brain, you would not live for too much longer after that, right? Because neurons don't reproduce at the same rate as your other cells do. And so for many years, it was thought that the immune system kind of treated the nervous system very differently. And it was even thought that the nervous system didn't make immune proteins. But in actuality, the nervous system does make immune proteins. And the thing that interests me, professionally even, when I was in graduate school, is that these immune systems that work in the nervous system have nervous system functions. And I think that's really, really fascinating. So C3 is not really performing the same role in neurons. So then the question then becomes, so what is C3 doing? So C3 plays a role in synapse existence, right? So the synapse is the junction between neurons. So neurons have to communicate with each other, and they communicate each other through these structures called synapses. And so you might have heard this in, just basic neuroscience, that your brain forms these connections between neurons, and then as you get older, these connections get refined. So you start off with a lot of different neurons with a lot of different connections to a lot of different neurons, and as you get older, from when you're a child, your brain will actually trim those connections, trim the connections that you don't really need, right? And so C3, this immune protein that helps the immune system blow up bacteria, actually helps you to trim your neurons, give them little haircuts. So it's pretty interesting. So these are just different types of synapses. So C3 is something that helps these synapses get trimmed. So I wanted to pull up this figure to try to give you an idea of what this data look like, right? So in green, we have these immune system cells called microglia, and in red, we have structures of neurons. So these are immune system cells. Microglia are the resident immune cell of the brain interacting with these neurons in red. And Clockwork, since you have not seen this, I want to show you this data pretty much fresh to try to see what your reaction is. But first, I want to let chat kind of try to interpret this. So here we have in panel E, the normal mouse neuron called wild type C3. And in red, we have synaptic material. And in green, we have microglia, these immune system cells. And so in here, we have a zoomed in version where you see this red dot inside the green cell. And when you knock out C3, the immune system protein, you find that the microglia no longer have these red packets of synaptic material. So chat, looking at this data, what does this say about the role of C3? What does C3 do to neurons? Don't worry about CR3 up here. Give you a second to think about that. What does C3 do and how does it do it? Kind of teased this idea in the previous slide unintentionally, so it may give you a little bit of a hint. So what does C3 do?
If I could add to the hint, since I'm gearing up for you calling me out, I think real quick reminding us that the red is present in when C3 is knocked out, like C3 is inside, right? So I'm just making sure I'm reading the data right, that's all.
So C3, this is a mouse with regular amounts of C3. And then on the right here in the knockout, it's a mouse without this immune protein. A genetic knockout.
Gotcha, yeah.
Yeah. This is kind of tough because it's kind of hard to see. But in the wild type, you have the microglia containing these synaptic materials, and in the knockout, the microglia no longer have that synaptic material. So, Clockwork, to put you on the spot, what does that kind of say to you about the function of C3?
Well, I can use one more piece of information, I feel like, that is from a previous slide that's not as obvious that it's a hint as you might think it is, but C3 is, just to make sure I'm not talking completely wrong here, C3 is part of a surface protein, right? Like it's part of, on the microglia, like the C3 complex is part of the C1, it's like, it is a part of the C1 complex signaling, right? Like C1's a surface protein, it connects to C3, C3 does the signaling inside the cell, so on and so forth, right?
I think you don't need to know that. You don't need that as a hint.
Either way, so when C3 is knocked out, we're not seeing the synaptic material present in microglia. Am I interpreting the data correctly?
That is correct. That is the correct observation. So what might you conclude?
So C3 is then critical in terms of allowing microglia to actually take in synaptic material. And since we know that microglia are part of synaptic maintenance, C3 is part of the pathway that manages managing synapses, to not use a recursive statement.
So this immune system protein that blows up bacteria in the immune system, in the brain, it actually helps microglia chew up the synapse. So what we see here in the wild type is that a microglia has successfully eaten a part of a neuron. And you need that microglia to eat that part of the neuron so that you have the correct connections in your brain. If you don't have this immune system protein, the microglia actually can't tell or don't want, quote unquote, to anthropomorphize this for a second, to chew up those synapses. And so you get a mutation, a lack of pruning. So you don't have that ability to trim the connections between neurons if you lack this immune system protein, which is insane to me. If you see here on the right, we have the engulfment percent. And so in wild type C3, you have a normalized value of 100%. So whatever microglia eat synapses, that's normalized to 100%. And we actually see that when you knock out that C3 protein, the knockout animal only trims about half of those synaptic connections as measured by synaptic engulfment. Okay, so there are neuron munchers or brain cleaning service, pretty much. And so these microglia need this immune system protein in order to do their job as brain cleaning service, more or less, which I think is insane because, again, when we grew up, we were taught that the brain and the nervous system and the immune system didn't interact. But here we are in the brain making immune system proteins, and it's helping us actually form our brain connections correctly, which I think is just nuts. So that was my protein story. I tend to like protein stories that have conclusions that are unexpected. It gives me a hit of dopamine when I find stories that go, oh, that's surprising. And so it was very surprising for me to learn that the brain was using the proteins of the immune system to do brain things. And I think that's really, really cool. I want to get your reaction to that, Clockwork. Does that kind of make sense as to why I find that story interesting?
For me, it's really awesome because the way we were taught was A, the immune system and the brain don't interact, and B, we were simultaneously taught, yeah, the brain cleans itself. Like you go to sleep and your brain prunes your synapses, fixes some things around, and that's kind of one of the one of many reasons that sleep is so important is because you need to allow yourself that time for your brain to rewire itself a little bit so that you can form longer term memories and all the other important consciousness stuff. So for me, it's like seeing the mechanism and understanding how it works as well. Another thing too, just knowing a little bit more about what people have been using to study structural biology. It's also seeing how C3 plays a role in different kinds of development. Like if you knock out C3 for whatever reason, certain mice don't have age-related hippocampal decline as well. What does that mean? Don't worry about it. I'm just thinking back to things I can remember, right? So that's what I love about it as well, is when we do these kinds of studies, there are so many proteins in the human body, in any living system, and it feels overwhelming when you're starting to learn the life sciences, because there's like 60,000 different interactions to memorize, which is not the correct number, but it's not even close actually. But the cool thing is that because we are able to isolate and knock out individual proteins, we have been chipping away at this gigantic complex system for like the past, I would say, 50 years at a wild level. And so therefore the proliferation of our understanding has expanded so much. And that's the thing that I love about this. This is a classic knockout study. And it's one of, tens of thousands that exist in literature, even in open access papers, things that you can go learn about right now. And that's what I love about it is, it's unexpected. We've finally discovered how the immune system interacts with the brain, which is really important as we begin to understand autoimmune diseases, the autoimmune contribution to certain neurological diseases. Just talking, thinking about how the immune system affects things like multiple sclerosis, right? What I love about this is seeing just how we can begin thinking scientifically and understanding, okay, I want to solve a certain problem. I want to cure a certain disease. I want to understand it on a protein by protein level. I'm going to knock out those various proteins, and I'm going to discover what those proteins do and how it contributes. So that's what I love about this, is seeing the process by which we see how signaling plays out. For me, the next thing, I would love to know the pathway of how microglia discern which synaptic connections are the ones that are marked for eaten and which ones aren't. But that is, of course, again, a very complex part of more advanced biology.
Yeah,
Love talking about.
I have a little bit of an answer for you on this very graph. If we take a look at these panels that we kind of ignored, CR3 is the C3 receptor. So if we see that the CR3, yep. So on one end, there's the C3. I believe C3 is the one that's expressed on the neuron, and I believe CR3 is expressed by the microglia. So the synapses that need to be removed are tagged with C3. How they're tagged by C3, I'm not entirely sure. But the microglia can kind of sense, they can kind of, when they find C3, they understand that that's a synapse that needs to be removed. And learning about the brain in that level of detail, again, and I think this kind of gets across to Clockwork what you're referring to. Biology is so cool. You don't have to understand everything. You don't. There's too much biology to understand. It's intimidating if you want to learn everything, but you could just crack open a paper, provided you have the education to actually make sense of it, and learn something incredibly cool any day that you are a scientist. And that's really, really neat. If there was so there's Sorry, there's just a chat question. If there was too much C3 in the brain, could that lead to excessive engulfment of synapses? So that's a very interesting question. I'm sure there's research out there where they overexpressed C3. If I had to guess from my memory, the answer is yes. If you make too much C3, I believe, and I could be wrong, but I believe microglia will chew up too many. So that effect is bidirectional. Sorry, Clockwork, go ahead.
No, I actually forgot what I was going to say. Like, that's the thing I wanted to get into as well, like trying to talk about the other side of it. So much respect to chat for asking the question I was going to ask. Love it.
Yeah, it's cool. Very, very cool. So let's put you inside the shoes of a biologist, chat. So we talked a little bit about the protein stories that we enjoy the most, and I want to try to challenge chat to think about how one could study the function or the structure of a protein that you don't know anything about. So this is another regular feature of my streams where I like to put chat in the position of the scientist just to work out those critical thinking abilities. So how do we study proteins of unknown structure / function? And there's a bit of a hint because we talked about some of those approaches before. So A suggests the first isolated. I mean, that's definitely worth doing. There are certainly many tricks that you can do to isolate the protein, but then how do you specifically study it? Remove the protein and watch the fireworks. Very good, Mr. Fox. That's what we did in the C3 paper that we just looked like. My idea is that we try different interactions between different proteins and put them in different situations. Yeah, so you can definitely take this in vitro approach. So this is in a dish or a glass tube and purify the protein and make it do certain things. And that's heading towards the approach that we will be taking in this upcoming study. X-ray crystallography is a great method by which we can understand the quote-unquote real shape of a protein. But how about we go a little more low-tech than taking a picture? Let's say we have protein A and it interacts with protein B. How do we study that interaction? What might we want to do? And think basic. Think just binding. Think about the fact that all of biology is just molecules banging into one another. How do we study the specific interactions between different proteins? What's one approach that we can do?
Could I take a crack at kind of guiding folks towards a way of thinking about it?
Yeah, go ahead.
So, like, I'm seeing a lot of really great techniques in the chat, right? But I want you to go back to the very beginning of this lecture, and I want you to think about sort of exactly how we get proteins in the first place, right? Like, if you had just like a magic box, and you could do anything to a protein structure, what would you do to it to see what it does? I don't want to get any deeper than that, but like, imagine you're not even thinking about the actual machines you would use. If you could think in the abstract and you wanted to see how two different proteins interact, what would you do to either one of them if you were a scientist with access to actual magic?
Yeah, I think that's a good hit. Yeah. If we could have access to the protein, how might we want to study it? And I think there was an answer in chat: remove it and watch the fireworks by Mr. Fox Tech. That's close. Removing it might be a little extreme. Certainly we did that with the C3 story, but if we want to study how proteins interact, what could we do besides remove it? This is more of an engineering kind of mindset. I think that's maybe a good hint. Think like an engineer. If you wanted to study the structure and function of a protein, how might you study that? How would you study anything? And this doesn't even have to be biology. What could you do? Break it down to simpler parts. So, that's definitely true. I'm actually going to borrow a piece of that answer. Breaking it down to simpler parts is definitely an approach that you can take, but there's actually an even simpler approach, and that's just break it. It kind of combines Mr. Fox's answer and A's answer, where removing it will definitely allow us information. Breaking it down will also allow us to get information. But if we want a very basic approach to understanding proteins, we could just break it and see, we could just hit it with a hammer, for instance, and see how that changes its ability to affect things. So all good answers that are relevant, but I'm trying to guide to one specific approach. So imagine it's 1997. We're starting to understand how insulin functions. So for those of you who don't know, insulin is a protein that you need to have your cells appropriately take up glucose. If you have dysfunctions in your insulin machinery, then that can lead to conditions like diabetes, which is no good, right? So it's 1997. It's some number of years ago. Where our understanding of how the system works isn't quite as good as what it is today. So scientists at the time were beginning to understand how insulin functions, and it would be really good to know how insulin interacts with its binding partner, the insulin receptor, for things like medicine and engineering, right? Because even today, we still don't have a reliable treatment for diabetes beyond some basic life maintenance kind of stuff. And so I kind of teased this answer in the previous question. There's something that you would like to know about insulin, the insulin receptor, is how do the insulin receptor and insulin bind? And actually we have an answer in the chat from X-ray crystallography would be pretty good to see visually how these proteins interact, but there's another layer of information that has to do with the primary structure, and that is understanding which parts of insulin are important for binding to the insulin receptor. So that leads into the paper that I chose to discuss today called Alanine Screening Mutagenesis of Insulin. And so this is a paper that you can get open access, link in the description, where we're going to be talking about an elegant way, an elegant method by which we can study protein. And elegant I use very cautiously, as Clockwork and I will talk about later, whether or not
I was about to come
elegant.
in. I was about to come hammer down.
Yeah.
How dare you call an alanine screen elegant?
Alanine screening is intellectually elegant. I think
Yes,
I can say that.
intellectually elegant, but as somebody who was raised by lab people, this is anything but.
So let's talk about alanine screening. So we talked about primary, secondary, tertiary, quaternary structure. And something that scientists can do is take the DNA sequence of a certain protein and change that DNA sequence one amino acid at a time, or one triplet codon at a time, to sort of scan the entire protein by changing each individual amino acid to an alanine. So alanine is your very vanilla, boring amino acid. Its R group is just this methyl, CH3. It's got nothing special. It's not even glycine, where it's at least flexible. Alanine is just your average, ordinary, vanilla amino acid. And so what we can do is we can take this string of amino acids, the string of amino acids that makes a protein, and in different systems, mutate that protein one amino acid at a time going down the line. So T, oh God. Do you happen to remember? Oh, tyrosine. No, tyrosine is Y.
Tyrosine's Y. Is this theanine? Oh,
Threonine. This is threonine.
Threonine. Jeez. Okay, hold on.
So,
I need to pull up. I literally have the same notes I had like a certain number of years ago when I was studying this. Just to make sure I would never forget it.
No, it leaves the memory quite quickly. So up here we have the normal protein sequence. Threonine. This is glutamic acid, I think, E. And then lysine, asparagine, aspartic acid, I think, is D. Watch me be wrong, but basically a string of amino acids, and you can substitute each amino acid one by one with this alanine. Basically the opposite of proline. Yeah, pretty much. Proline is exciting and kinky. Alanine is kind of boring and vanilla. And so what this allows us to do is we can compare the binding activity of the normal protein and compare that to the proteins that we mutated one amino acid at a time, just to figure out which parts of the insulin protein is important for binding to the insulin receptor. So intellectually, this is a very elegant approach, maybe not the most practical, but it is very satisfying in a certain way.
And at the time, it was also like the best way to do it too. Like cryo-EM and like different forms of microscopy were kind of fidgety, right? Like you could do it, but you had to do it perfectly. Whereas this is a really great way just to like lock in your variables.
Mm-hmm.
It just is very labor intensive, which is the only thing I got into.
Yeah, so I think we kind of tease this next follow-up question: what are the potential pros and cons of this approach? The pros is it's very elegant, very, very elegant, a very elegant way to step through a protein and systematically change the protein one part at a time. It's the engineer's approach, right? If you're an engineer, then you love breaking things and then fixing them. And so this is a way where we can break the protein a little bit at a time to figure out what every part of that system actually does. And as Clockwork had teased, this is very, 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 very labor intensive because proteins are quite long. Even insulin is not insignificant. In fact, the authors don't mutate everything in insulin, just the part that they think might be relevant for binding. So this is basically throwing graduate students into a meat grinder because each one of these systems, each one of these proteins that are changed, that's a lot of work to do. That's a lot of bacteria to grow, or yeast, or whatever expression system you use. It's very, very labor intensive, but it is simple to understand. So let's continue on. So Clockwork put together this wonderful GIF where you actually get to see the different mutations of the insulin that this paper uses. So this paper takes the insulin and mutates the insulin one amino acid at a time in the beta chain and alpha chain. And so you can see there are beta chain mutants and alpha chain mutants, and we can see that those individual amino acid changes actually does have quite a significant effect on how the protein folds. And what this paper is trying to get at is understanding what those changes to those protein structures have on its ability to bind to the insulin receptor. So here on the right, you have the very easy to understand GIF that Clockwork threw up. And on the left here, we have the actual figure from the data. So you can actually track which of these amino acids this paper actually cares about. And you can see up here is the normal amino acid sequence, and then down here we see which ones have been turned into alanines or A's.
And then to like really lock it in, audience, what you're seeing in the GIF, the little white parts are the actual spot where alanine is being substituted. You're looking at what I did to make this was I threw these structures into AlphaFold, Google's protein folding AI, got the structures out because here's the thing, it's actually hard to find these mutants in scientifically accurate cryo-EM data because people aren't just throwing useless insulin mutants under a microscope or anything unless they're like relevant to a specific disease. So these might actually not be perfectly accurate because they're computer generated, but it's good enough to illustrate what we're talking about. So the white is the exact spot where an alanine has been substituted, and then the rest of the chain. It's amazing how far away that alanine is, and you can actually see changes across the whole chain because of that.
Dr. Toto is in chat. Hello, Dr. Toto. Welcome to the stream. Another educational VTuber. Give her a follow. Welcome. Yeah, so let's talk about insulin binding assay. So so the authors made these mutations of insulin, and we're trying to test whether or not insulin still binds to that insulin receptor. And so, thanks for joining in. Dr. Toto has to go back to teaching. Yeah, thanks for dropping by and saying hi. So how are we going to study these insulin mutants? We're going to make these insulin mutants, these proteins of slightly different variety, and then we're going to purify the insulin receptor. So we're going to add radioactive insulin to the insulin receptor. This radioactive insulin will allow us to detect how much of that insulin is actually bound. And then we're actually going to chase off the insulin with insulin mutants and measure their competition, right? So this is a pretty important concept to understand when we talk about biochemistry, this idea of competition for binding. So here we have a structure, and in orange and green, we have the two, I think blue as well, the subunits of the insulin receptor. No, blue is insulin. And we have two molecules of insulin, this one in magenta and this one in blue. And so we're going to first preload this receptor with radioactive insulin so we can measure how much insulin is here. The radioactive insulin gives off a signal. And then we're going to chase off the insulin with insulin mutants. And so measuring that competition is going to allow us to measure whether or not the insulin mutant binds tighter or binds tightly or not as tightly as the normal insulin does. And so that's an important concept to get. So I'm actually going to do a check for understanding. So I'll write the question in chat. If the insulin mutant breaks binding, will you need more of it to chase off the normal insulin or less of it? So that's the question. I'll set up a poll. This is a part where you get to flex your understanding of the system. Preloading the insulin receptor with radioactive insulin, and then chasing it off with an insulin mutant. So if we break the protein by accident, or in our case on purpose, will you need more or will you need less insulin? All right. Yeah. Yeah. Okay. Yep. So chat, very good job. We need more. We need more of the mutant insulin that doesn't bind as tightly. Okay, so we're locked in. We understand how these assays work. Now it's time to get into a little bit of that data. Okay. So we're looking at the actual data of the binding assay. And so what we have here is in dark squares, we have normal insulin. And so when you chase off normal insulin with normal insulin, you get this kind of shape. And so when you chase off with B1 alanine, you need more. So this is open triangles, and A19 alanine, dark triangles. So on the bottom x-axis, we have the concentration of the chaser, so the mutants, and on y, we have maximum binding. So at 100, if you chase off with normal insulin, this is the shape. You chase off with these mutants, and you get this shape. So when we add the mutant insulin, we end up needing more to chase that normal insulin off. And as Chad has correctly described, that means that these mutants don't bind as well. So the highest affinity was the normal insulin, and then we have these two mutants, which end up dragging the graph to the right. And that drag to the right means that those particular amino acids in those positions of that protein are important for their interaction with the insulin receptor, which is very, very cool. Okay. They compiled all of their data together to give us sort of a map to describe which of these amino acids are actually important for the interaction with the insulin receptor. So down here, we see all of the different amino acids that were assessed. I think the study doesn't do all of these, but this is like conglomerate of all of the alanine screens that were done in this protein. And we have this nice graph. This relative affinity on the right here allows us to see how broken these amino acids, or how important these amino acids are for this interaction. So chat, it'd be good to sort of ask chat, given this data, and look here, right, relative affinity of insulin, which of these amino acids appears to be the most crucial for insulin binding to the insulin receptor? Down here, relative affinity at 100%. If your bar is down here, that means you're about as good as normal insulin. So which of these amino acids in which positions is the most important for binding with the insulin receptor? Font's a little small to see, so you could just describe to me which one of these bars. Give chat a second to think through that.
And one thing too to keep in mind as well, chat, if you look carefully, this graph is actually on a weird scale. Like it goes from 10%, then to 1%, then to 0.1%. So a lot of the action actually happens in the first kind of quarter of the graph, but it's really important to see what happens after 1% as well, if you look at that scale too.
Yeah, the scale is kind of nutty. Yeah. A19, A says A19, that's definitely true. Look, just one change all the way over here, changing this tyrosine to an alanine, one change goes from 100% normal binding to about 0.1% normal binding. This is an incredible discovery to find. This A19 amino acid, you could conclude, is probably very, very important to the insulin receptor interaction. And so I want to give chat a kind of chance to sort of hypothesize why this particular amino acid, like what is this amino acid even doing where it alone contains so much power. Given everything else here, like as Clockwork had mentioned, if you're at 1% here, like these are also really important, right? A2, B6, these are all still mostly abolishing this level of binding. But it's kind of interesting to use your imagination to kind of guess as to what this A19 amino acid is doing. So you no longer get this binding. What could A9 contribute to? Just thinking out loud. And I actually don't know the answer. So it'd be really interesting to poll chat to see what they think could be the answer. So A thinks that A19, this tyrosine, could create strong bonds. Certainly tyrosine could be quite important in that position for binding to the insulin receptor. But what other hypothesis could you have about A19's role in this interaction? And I'm wondering if Clockwork, you kind of know where I'm heading to in terms of a hypothesis.
Are you trying to reference back perhaps to the very beginning of this lecture where we talked about what each individual role of each individual amino acid and trying to see exactly
Oh, that's maybe a little too specific. No,
rats. Okay.
my idea is a lot more basic than that. So A is certainly correct that this amino acid could be very important for the specific interaction. Maybe it lies at a certain area that touches the insulin receptor, and maybe if you change that tyrosine to an alanine, that kind of screws with that interaction. But what other thing could be going on? Could be. And A's got it again. Folds the insulin into the correct shape. Exactly. That's what I was thinking as well. So it's certainly possible that A19 is in like an important amino acid in the binding pocket for the insulin receptor, that it needs the specific shape to fit in. It's also possible, although less possible, that A19 is important for the entire protein structure. So if it just loses its shape entirely, so maybe it's not specific to the insulin receptor, but rather the integrity of the protein altogether. Whether or not that's likely is kind of hard to say, because tyrosine isn't, it's not cysteine, right? It's not methionine, it's not disulfide bridge forming, but it could be important. And it's important to keep in mind that there are different hypotheses for the same data. All right, I think that might be the end. Clockwork, do you have anything to... Oh, these are some gifts that Clockwork let me use. Yeah, that's the end of the protein stream. Are there any questions in chat about proteins? Questions for me or Clockwork, anything that we talked about? Especially since Clockwork is here, and Clockwork, you don't really stream. If you have questions for Clockwork,
You get one chance.
time, yeah, you get this chance.
It's actually upsetting because I had everything set up for a GIF showing insulin on the insulin receptor, specifically comparing wild type insulin to the A19 mutant. And then I rendered everything, and it just did. The renderer was like, "I'm not doing that one," and then I forgot to finish it. So I'm sorry we didn't have that here to kind of show where tyrosine hangs out in the binding pocket as well. But there's several factors that the A19 actually contributes to. I'm the only person who can see it right now because it's still on my computer and not in an actual GIF somewhere. So my bad, y'all.
You can also just send me a screenshot through Discord, and I can just put it on my OBS.
Oh, that's wild. Okay.
Yeah.
Challenge
So
accepted.
yeah, it won't be a GIF, but that still is better than nothing. I want to.
It also won't be rendered properly too. It's very what is it called? Janky when it's not in its rendered state.
Oh, gotcha.
But that's not a big deal.
Yeah, so I want to thank Clockwork for putting in the work. I always make sure that I communicate to my collab partners that they don't really have to do all that much work for collabing with me, that I'm very okay with doing most of the work myself. But I mean, so far every collab partner I've had has been very generous with their time, and part of that generosity is doing things like rendering these insulin mutants, which is very cool. And Clockwork, when you get back,
I'm right here actually. No,
okay.
I'm just making sure I'm lined up, and then I have to turn off regular insulin. Goodbye. Hello. You come here, you get in there. Good. Screenshot shotted. And that's another thing too, audience, that I want you to kind of keep in mind is the just collaborative spirit that's here in the life sciences. No matter what, you're going to see that in terms of precisely like when you're thinking about that phi, like I think that's just kind of the spirit of the science, right? You see folks in life sciences kind of drop everything for each other a lot more. What's going on with this? Why am I not allowed to take screenshots anymore? What is happening?
I got your thing.
Okay, you
So
got one of them. So you've got wild type insulin in the binding pocket, and I'm trying to get you A19 to show you precisely where it hangs out. But
gotcha.
how I usually do screenshots is eluding me. Am I in here?
Interesting.
Okay. I hope that's it. Is that actually happening? It's weird because I just did this screenshot, right? And I'm doing the same process for another one. It's like, no, I don't want you to do that.
I've learned that anything that does rendering is just it's just black magic.
Yeah.
Having worked with Adobe Premiere for so long, I'm not surprised that it's messing up for some reason.
Okay, there we go. Okay, check this out. So you've got wild type insulin on screen now, and I'm going to get precisely where that A19 tyrosine is. You can kind of see it. It kind of occupies a very critical spot of the whole process. But Phi, what you did was really brilliant too, because you talked about how potentially it could affect how the insulin folds as well. And one thing we don't really cover is that insulin, when it's originally encoded, it actually has a couple of the A and B chains aren't the whole residue, right? Like insulin's actually several hundred amino acids long. It just gets trimmed once it goes from protoinsulin to insulin, if that makes sense.
Yeah, so we can actually compare these side by side, which is super neat. Yeah, so this alanine here, this white part here, does appear to be pretty important, it looks like, for the interaction with the insulin receptor. Comparing here on the left, this white spot to the lack of a white spot. So it looks like here that there's this little gap on the right between the pink swirly and the blue swirly, and it looks like that alanine might be getting in the way of that particular structure, which I think is very neat. So we got some questions for you in chat.
Let's go. What do we got? What do we got?
Yeah, what is your favorite immune cell and or protein?
I had to think about that a lot in terms of trying to enter into the Phi cinematic universe, right? It's actually very tough to say because outside of the neutrophil, a lot of your immune cells are they're pretty dramatic. Like my initial thought was like, yeah, I'll just I can say I'm Clockwork of the basophil, but the basophil actually contributes a lot to like histamine responses and other gross stuff. And I thought eosinophils were cool because they just have a cool name, but they also contribute to some unpleasant allergic reactions. So, honest to God, the neutrophil's like the goat in terms of like the phagocytes in your immune system. Macrophages are cool because I've been doing a video on tuberculosis, so I've been talking about exactly how tuberculosis just punks human macrophages. But at the end of the day, like I was raised like in an HLA lab, basically. So I'm always going to say, your CD4s, your CD8s, especially now that we're doing cool stuff like engineering T cells and B cells to fight like autoimmune conditions. So the answer is all of them. I'm sorry, they're all very cool. Immunology is we're such a cool spot in terms of studying immunology right now that they're all genuinely fascinating. Like all of the various cell types that power your immune system. The YouTube channel Kurzgesagt made a beautiful book about your immune system, which is really super accessible if you have the ability to find it. Even get it on audiobook is amazing. So highly recommend learn everything you can about this incredibly dynamic, incredibly detail-rich part of your system's biology.
The other thing about the immune system is, as a kid, having watched like Osmosis Jones growing up, it certainly is something that I really wanted to study growing up. An actual real immunology learning experience, you have to be ready for some pain.
I mean, most of it, here's the thing. Advanced biology is maybe one of the most painful things to study because of all of the interactivity that's going on. You're going to go through OChem and it's going to absolutely break your brain. And then that's never going to stop because you need all of that OChem to understand protein on protein interactions and everything else. But the cool thing for me about getting through those initial advanced topics, getting through the air quotes weed out courses, which that's not a term I'm super enthused about, but once you pass through that and like kind of trust your scientific intuition a little bit, once you get to the point where you're starting to really think interdisciplinary about biology, it really becomes so rewarding just seeing all the different connections. You spend all this time studying one part of one residue in one protein, and you find a similar amino acid pop up and do something analogous in another structure, right? And not even in protein systems, but in cell systems as well. For me, that's where the real reward lives. Like, if you want to see how I kind of think about that way, I have a whole series on photosynthesis where I connect like chloroplasts to the universe. And I think that's a cool way to sort of think about like why advanced biology is super rewarding. Because if you think about these things at the chemical level, you can kind of see the whole history of life on Earth a little bit laid out before you if you think sort of like at a geological time scale and think about evolutionary pressures. So
Yeah.
that's I
I,
don't even remember what the question was at this point. I just had to go on my tirade about
I
it's super
had
rewarding.
so
Study more.
there are a couple of things that I think are interesting conversations to have with a fellow biologist who I think our paths are somewhat similar. It's kind of interesting.
Biojournalist. Like, I
Yeah.
did study biology, but I did study biology at a liberal arts institution. And if I could go back in time, I would have told myself, hey, go with a pure STEM program. There's nothing bad about the liberal arts, right? Especially if you stay focused on the actual major you want. But the thing about a liberal arts institution is you will gradually be guided to where your aptitude actually is. And unfortunately, my aptitude has always been in writing and communicating these things, and not so much being an actual scientist, right? You understand what I'm saying?
Yeah, but that's kind of what I'm referring to, in the fact that like
Yeah,
I'm no longer a practicing scientist, but I think it's way more fun to share than it is for me to be the one in the lab kind of
very
grinding
much
it
yes.
out.
Oh, yeah, being in the lab, I mean, looking back on it, super rewarding, super fun. Would do it again. Give me a lot of money, and I'll just build, a histology lab. However, the guy who was doing that, the little baby Clockwork, was not super happy trying to figure out white knuckling through various PCR assays. Even PCR was hard, right? So just imagine doing an alanine screen, dude. Just like the amount of expression you have to do, the amount of precision you have to nail in. You have to spend $3,000 on radioactive iodine, and then hope you don't waste that whole assay for your insulin. Like, that whole process is just,
And cloning is kind of a
oh yeah, that
mysterious
too. Yeah,
process on its own. Yeah, so
yeah.
on to go back to a conversation topic that I think is quite interesting. It's the I'm an instructor, so I spend a fair bit of my time thinking about the meta of learning, as my audience may know and be aware of. And you said that you did not like the idea or the referencing to weed out courses, and I kind of want to hear a little more about that, as from the perspective that I have as someone who does do education professionally.
I
Oh,
think
yeah.
no, go for it. Sorry. Um, I think what you're going to come back at me with is valid. How we need to sort of make sure that people are prepared for the complexity in these courses. However, I think I have experienced a lot of folks who take pleasure, not who get a sense of satisfaction of watching folks leave the discipline. And I feel like the way a lot of our educational system exists, a lot of people need more second and third chances. Like, if you were able to take like 2 to 3 cracks at even let's just say organic chemistry, right? Until you like fully understood it and had like sort of the right combination of tutoring to really help it crystallize for you. That creates somebody who is able then to move on and do more science. And I think the world is a better place when more science happens. Like, I think most of the 21st century being a good place is because like the polio vaccine and eliminating smallpox. If we had more people doing that kind of work, you do more work in an area, you develop more pieces of it, people make more connections, complexity rises, and you get better things. That's not a perfect analog, but when I think about weed out courses, I think it's important that suddenly biology goes from cool to stressful if you're studying OChem, which is what it's designed to do, because you need to develop your scientific intuition. But calling it a weed out course makes it seem like you want folks to fail, which, when in reality, what I want is like 10 times the number of doctors we have, among other things, right? I think that
Yeah,
would be,
I
yeah.
think the perspective is interesting. And as an educator, I think that perspective is valuable, in the sense that I really like what you said about some people needing a couple of chances. Um, and I'm wondering what universities can do to formalize that kind of idea, right? Because from my perspective, weed out classes, it's not even about developing your scientific thinking, because as I'm not an authority on university education, right? Like I teach at a university, but that doesn't mean I'm like an expert on the meta of teaching at a university level.
Feel an
Um,
expert on the like specific circumstances of you teaching at a university, whereas
exactly,
I've
exactly.
only ever like I've only ever taught the high school level, and like I've tutored pre-med students, but that's barely university level,
And
right? Like you've actually been in the system.
it's a pretty interesting perspective because like I would love for people to have all the chances that they need to nail something down. And also in terms of I don't even agree in the sense that like weed out classes are even useful, even in the content, right? Like you say that the weed out courses develop your scientific mind. I actually don't even know how much organic chemistry contributed to the way that I think as a biologist. Rather, I think from my perspective, my personal experience being a student in school is that they were more like uh skill checks, like discipline checks, maybe, which maybe disadvantages students from certain backgrounds. Or do you kind of get what I'm getting at? It's something that I'm just like formulating in my head right now because of what you said about some certain students needing a couple of different chances.
No, I hear
uh,
you. I hear what you're saying too, because it's one of those things where eventually, the rubber will meet the road, and you will have to be in a lab and be trusted to use those extraordinarily delicate and expensive resources in a way that helps advance our scientific knowledge or advance the goals of a lab. And if you've kind of like had a gentle hand the whole time and don't really understand and have a respect for the incredible power that some of these systems have, just like thinking about what a PCR machine does and just like the amount of information it is, resynthesizing just so you can like study it with your feeble human mind, is incredible, right? And so I understand what you're saying too, and I respect that position. You asked me a question and I just kind of gave you an answer, but you're right as well that like you have to instill it's not discipline in like the traditional sense of the word, but you have to instill that procedural way of thinking that gets you through a scientific problem. You know what I mean?
yeah,
Is that yeah.
it's like this is why I think it's interesting to chat just casually about, just because I've been seeing a lot of discourse online, and it's been in my Discord as well, this kind of pushback on like brute force memorization as a biologist. For those of you who are interested in studying biology, there's a fair bit of brute force memorization that you're going to have to do, and that's just what you're going to have to do if you want to study biology. There's no getting around that. But there was this thread on Reddit that I've been thinking about people complaining that there's too much to learn when, outside of their direct interests. And as an educator, I mean, I'm not speaking for all educators. Maybe there are people who agree with that. But the idea is people, I think, construe college in a way that is maybe not very accurate. College is for learning how to tackle hard problems and not really learning information, as weird as that sounds. I mean, certainly the content that you learn in a biology class, if you want to be a biologist, is important, right? You need to know what the mitochondria does, but you also need to teach yourself how to teach yourself about the mitochondria, which is exactly what college is about. And there was something else that you had brought up earlier in conversation that I wanted to circle around to, but I've kind of lost that.
It's the classic biology conversation where we end up talking about maybe I would say 70 different topics and we want to focus on one of them, but we always forget like which one it was exactly. That's half the fun when you get this much into the weeds.
Yeah, and like I think, yeah, I almost wonder if chemists feel the same way, because for biology and for history and for art, I think there's like this rich tapestry that all connects in this like wonderful, interconnected work of art that we call life. Um, and art from different periods can connect historically to other art from other periods, and of course history is just one long story of humanity, right?
Mhm.
But I wonder if chemists also feel that level of interconnectedness between what they learn, because from what I understand, and this is maybe a huge overgeneralization, I'm sure chemists they, if they're in chat or they watch the VOD, will correct me. But it feels like chemists are very focused on their one problem, synthesizing this thing, getting this chemistry to work. And I wonder if they also have the attitude of chemistry as a broad tapestry. It'd be kind of interesting to find out.
I think, it's one of those things where, based on the folks I've talked to, like in calmer moments, I'm sure you have that sort of thought there, but like chemistry, physical chemistry specifically, you really do have to drill down into your like specific thing you're trying to do. Otherwise, it's very easy to get distracted, And it's one of those things where the synthesis itself is the goal, just thinking in terms of pure like reaction yield or whatever. But I think everyone thinks that way. I think it's a very human thing to do, to try to, whatever you're doing, you want to connect it to a broader story. Like I want to be able, yes, I'm studying hemoglobin, but I'm also studying like how like this affected my place in the universe, right? Why am I here in a nondescript part of the eastern seaboard of the United States? Can hemoglobin teach me anything about that? Not really, but it can add to my understanding of the world, which helps me understand precisely where and how I am.
It would be kind of neat to talk to a chemist and wax poetic about spirituality and the practice of science, right? Because physics, there's the obvious connection of cosmology in terms of like reflecting on why we're here, and biology is very obvious too because we deal with evolution, things like that. Chemistry outside of biochemistry, and like I wonder if they have that equivalent feeling of the human experience in their study. Or how that manifests.
I do too. I mean, I'm wondering. Like, it's funny how we both can't come up with an answer. Like, we can both think of like how physicists think about it. I think one big problem is that when you are in the life sciences, when you're a university instructor, and I just talk to them basically. A lot of those folks don't end up talking to a lot of like physical chemists like a super lot. Like you kind of, we're in a period of specialization where it is very hard to have those interdisciplinary conversations at that level when you are, advancing the scope of human knowledge, unless like you make an actual breakthrough and your discoveries get shared widely. I also think a lot of like physical chemistry ends up getting eaten by the great wall of engineering. Like a lot of people aren't getting into chemistry to study chemistry. They're getting into chemistry to get into chemical engineering and to do all that fancy stuff, right? Is that another way of thinking about it, or am I generalizing too hard?
I don't really know because I don't really know too many professional pure chemists, because obviously due to what I do, most of the chemists that I know are also doing biology in certain ways. But yeah, it'd be cool one day to find a chemist and chat about that kind of thing.
You guys gonna turn into like a huge source of just like an endless stream of like physical chemists who come in here just to like be a part of your chats from now on. Like for some reason, the beacons of Gondor are lit, dude.
Man, that would terrify me, man. Chemists and structural biologists, frankly, terrify me as well. Not to say that, it's the fact that like I have like scars from talking about or failing that portion of my final exam, the X-ray crystallography part. It was pretty traumatizing.
Did you fail theory, or did you fail like the lab practical?
I failed theory.
Yeah, I failed the actual like NMR phase of my OChem 2 file. Like I just completely botched the data. So it's funny how we had the opposite experience.
Yeah, like doing the NMR I thought was fine, and even understanding like what NMR is theoretically was good, but like I don't know if you've had this experience, but on my final exam they gave me like the photo 53 or whatever, like the initial
photo 51, photo 52.
Yeah,
52?
photo 52 or 52. Whatever it is. The X-ray crystallography photo that you can deconvolute to draw a picture of DNA. So what they did is that they took that level of data, just that wonky shadow, and then they wanted us to draw what protein that blot reflected.
Wait, no computer, just like a raw X-ray blot. You had to look at each individual dot and be like, that one's darker, that one's lighter, and then figure out where everybody was, basically. That's a nightmare.
More or less. Although it wasn't looking at individual dots, because, it was just like, what is the symmetry? What is the spacing between this, et cetera, et cetera. And I studied so hard for that exam. I couldn't study it fast enough to be able to go from an X-ray shadow to an actual functional structure, and that's kind of like, dang, I guess I'm just not like cut out for. And that's the other thing. That's another interesting thing about like picking a discipline. I was just like, I'm not good at it, so I'm not gonna try.
Well, it's
Which
also
is
one of those things like, do you need like at this phase with computational biology being as good as it is, do you need to be super good at that? Like,
I
it's one of those things
think
where like, I do I need arithmetic to like do accounting? Like calculators exist, you know? Like, yes, the theory is good, but like the tools we have are excellent enough to carry you most of the way,
I
right?
think the idea that I could not arse myself enough to study informed me that that lifestyle was likely not where I wanted to go.
That's really important. That's really good
Yeah,
information to have. That's kind of what me and OChem 2 figured out as well. Like, I knew in any exam environment I thrived, but when the final exam it happens in a laboratory, like I completely just short circuited for some reason. Like I knew NMR back and forward. It's also like a very easy graph to read. There's some memorization involved in knowing, where each hit's going to correspond to whatever structure, but it's like that's not part of the exam anybody else failed. Like I passed the course still, aced everything else, but just like being in that environment, that also told me, oh, okay, like being in an actual lab probably means a lot of high stress situations where I have to very quickly do these like procedures and do these practicals. Probably not gonna be where I'm going to be successful if an exam kind of freaked me out too much, you know? Like I don't
yeah,
think I could handle high stakes biology.
yeah. No, that's very fair. Where money and reagent are on the line. All right, so we've had a stream. It was very fun getting to chat with you, Clockwork. I will be wrapping up now. I'm gonna go back to the beginning of this presentation to do my plug one more time. But in the meantime, do you have anything to plug, Clockwork, coming up?
So I'm excited. Personally, I'm going to plug what you're about to plug, which is your video on prions, which is going to launch tomorrow morning. Very excited to get your take. Just on you've been doing a lot of really great series in terms of like different diseases and different sort of dysfunctions that happen. So I'm excited to see your video this Saturday. I'm also excited that I'm finally putting a video out after years of being in development mode next Friday. I'm covering eukaryotic DNA replication, which is just it's been a very fun topic to write and animate about, and I'm so excited to kind of take you guys through it. We talked about the central dogma during the stream today, and so I'm going to take you guys through the ground floor of that process, which is copying your DNA so you always have it ready and available for when your body needs to transcribe and translate DNA to RNA to proteins and to do, cell stuff.
Yeah, and so there's like that. I think that's going to be particularly interesting from a protein point of view because that entire process is so highly coordinated. It's pretty amazing. Before I sign off here, I've been keeping my Discord semi private and I've only been copying the link for people who stay till the end of streams just so that I can control kind of the tide as I slowly get this Discord server, regulated. So if you're in chat and you want to hang out for a hot second and you want to join the Discord where you'll get to hear, my thoughts on biology or reflections or additional information, stuff like that, or just hang out and chat, I will leave that link in the chat. Go ahead and copy that for you chat members if you want to join. No pressure to. Although that one, yeah, see if that works if you want to join the Discord. You'll also be able to give yourself a role where I'll be able to ping you whenever I go live or make content. Yeah, so watch out for my Prion video tomorrow. I've put a lot of blood, sweat, and tears into that video, and I'm really glad to be done with it. It's really been, I mean, it's not quite as long as what you've been building up to, Clockwork, because I think if you were to contrast our stuff, mine is a lot less effort, but I like to move quicker in terms of like Clockwork. You put in so much craft into everything that you do, and I'm
Well,
like,
think about it though. Think
this is
about
a
it though.
minute.
I have to. You have a lot of foundational knowledge to fall back on, and you can confidently do that. I have to lock in every single bit of understanding I have because I also make the mistake of talking about the cutting edge of research, right? So if I just low effort did that, I would make I can't even imagine the amount of mistakes I would make. And that's one of the reasons this video in particular took much longer than I thought, because I actually did make one catastrophic error in terms of how DNA is primed. That had to be, every single aspect of the video had to be completely corrected. So, it's one of those things where your expertise compounds over time. So, your videos are high effort. It's just you put in the right effort earlier. You know what I'm saying?
I suppose you could say that, but there's no way that you're getting me to open up anything that you use to make all these structures. I'm allergic to that level of detail.
You would have so much fun in Blender. Get molecular
Oh,
nodes,
I don't
download
know.
Blender, go through a structure one residue at a time. Like it's a good place to play, dog. I mean, I'm not telling you how to live your life or anything, but I think you'd be surprised how intuitive it becomes once you're in here a while. Of course, I'm speaking as a convert here. But to really what you said, like I have the exact same situation where like I put in so much work to these videos, and I literally forget 90% of what I said within minutes of publishing. Like it's just gone forever. And like it's so satisfying to have that information not in my brain anymore. Feels
Yeah.
very sad, but at the same time, that's just kind of the reality of putting this much effort into talking about a subject in a way that is understandable and digestible for a broader audience.
Mhm. Uh, I think that's a pretty good place to leave off, honestly. Thank you so much, Clockwork, for joining me here in this protein lecture. I had a lot of fun and learned a lot. Yeah, go subscribe to Clockwork, and now I am going to head to the ending screen. Bye, y'all. Oh, no, this is the angry preset. Which one is the preset? This one. Bye, y'all.
Be well. Thanks so much for your time.